Well, this project was, it's quite an old project already. It's been going for a few years, and it was initiated by Guido Capitani. I talked about this a few years ago in another 3D edition about the first phase of this project. And basically, it's a kind of a second phase of it, and it's mostly about evaluation, prediction of quaternary structures in protein crystals. So we heard a lot about protein interactions this morning, and it's important to see the primary data where these protein interactions come from is the crystallography. And as Ilan just said, that's kind of a very artificial context for the proteins to be in. And one of the and actually, the data is still coming a lot from crystallography. We have 90% of the PDB still from diffraction, X-ray diffraction in crystals. And the problem there is that there's kind of ambiguities of how the quaternary structure of the protein in solution was. So there's this kind of problem that you don't know what's the actual quaternary structure that was in solution. So this is a 2D depiction from inspired in an Escher drawing. How it looks like more in real life. This is actually a protein that was solved in the group of Guido Capitani. This is a heterotrimeric protein, and they had the actual problem that it was difficult to see in that crystal whether that the third interface in that trimer, whether that interface was the biological relevant interface in there was that one or that one. So that kind of ambiguity is always present in any crystal, and you always have this problem where you want to classify the crystal contacts from the biologically relevant interfaces. So going to basics about proteins in solution, there's the a long time ago, more than 50 years now, there's the famous paper from Wyman and Chargeux that presented the allosteric model, but also presented a very simple reasoning of how proteins associate in solution. Based on really very little data at that point, because they knew very few protein structures, they reasoned that there's only two kinds of possible interactions for proteins to associate in solution. First one being the isologous one, where there's an interface that is used by both partners in the interaction. So both sides of the interaction use the same interface. That's called an isologous interface, or face-to-face. Then we have another kind of interaction, which is the heterologous, and there's two interfaces there. So the interaction happens through different interfaces in each of the partners. That's called heterologous or face-to-back. The issue with that is that there's kind of open interfaces that are exposed to the solution, which means that you can then keep associating other proteins to that complex. With that, we see that from isologous, we have, when isologous interaction happens, this kind of complex happens, and that complex must be in a two-fold symmetry interface to be able to be isologous, and you have directly a symmetric assembly which is closed. There's no open interfaces there. Then in the heterologous case, there's two possibilities. One is that the proteins associate kind of linearly, and they form some sort of fiber, so they keep associating, and there's no kind of a stable monodispersed kind of protein solution, but rather a fibril. And the other possibility is that the interface is kind of bent, and it curves around, and it goes on around until it meets the first protein, forming a cyclic symmetry. And again, it's a closed assembly, so that gives you a soluble protein that is stable in solution with a single kind of species in solution. So that means, after all, that a very important concept that Mono and colleagues introduced is that oligomers must be symmetric, and by that I mean homomers or heteromers. And this is a beautiful figure from the Levy and Teichmann paper, where they present all the possible point group symmetries, cyclic, dihedrals, and the tetrahedral, octahedral, icosahedral. And importantly also, this applies, as I said, for heteromers too, and the consequence for heteromers is also that they are not only symmetric, but they must have even stoichiometry, which is because of the symmetry. And actually, this can be seen in the actual data we have in the protein data bank. We have 96% of the annotations we have are for symmetric complexes. And there's some evidence that some of that 4% might be wrong. So not all of it, but there's some evidence for that. There are exceptions, of course, to the symmetry, that it's possible to break the symmetry in a protein complex. Some mechanisms to do that are pseudosymmetry, conformational flexibility, or steric hindrance. A beautiful example of that is the vitamin B12 transporter, where there's it's a transmembrane protein, and it's three kind of domains. There's the 2D chains that form an isologous twofold interface. There's then the C domain, the same story here, an isologous twofold interface, and then there's a single molecule that caps that C domain and binds symmetrically to that C dimer. So in the end, you do have here a non-uneven stoichiometry. If we extract that F domain, you can see here beautifully a twofold interface internal in the chain. This is pseudosymmetry. This is actually analyzed by the CESIM program of Douglas Myers and Spencer Bliven. You see here basically that the two, even though the sequences are different, they form sort of a pseudosymmetric interface that really can bind to this exact same partner in both sides. Having said that, it's important to say that the asymmetry, observing asymmetry in a crystal, doesn't mean that there's an asymmetric assembly solution. And this happens sometimes that there is a certain heterologous interaction in the crystal that doesn't really, is not really extended in the crystal, but is rather capped by other molecules around. And but here you see the same problem as we saw before. If you extract that to solution, you will eventually, it will be difficult for that to be stable because there will be other aggregation on those open interfaces. So if you see something like that in a crystal, it's important to really do all the extra experiments to confirm that that's actually a real thing that happens in solution. It's not some kind of metastable state that it's captured by the crystal. So having said all that, is now introducing how we are doing prediction of quaternary structures. We first are doing scoring of pairwise interfaces. We basically, from the crystal, we enumerate all the possible interfaces that we can find. Then we can do scoring per interface. So for instance, and the scoring is geometric in one part, but also it comes from evolutionary data. So we find multiple sequence alignments of sequence homologs that are close, evolutionary, something like 60% sequence identity to the target protein, and we map that conservation to the target protein, and with that we can find a differential score for the differential conservation of the interface versus the rest of the protein. With that, that's what we did some time ago. We did the EPPIC software, evolutionary protein-protein interface classifier, and this is kind of an output of the web server. Basically, in the end, for every interface in the crystal, we assign a biological, so assign it as biological relevant or crystal interface. We also, since the latest version, are doing confidences, which can be very useful for the final user. With that, then the second step here is to actually combine these interfaces to create a quaternary structure prediction. And for that, it's important not only to understand how the interfaces look like, but also the topology of the protein in the crystal. And that's how to do that. The best way is to represent the crystal as a lattice graph, where the chains are the nodes and the edges are the interfaces between the chains. This is how it looks like in 3D. And this is a periodic infinite graph, and this is a tool used a lot in mathematical crystallography. They are called crystal nets, and especially in relation to the small molecule crystallography. So if you see that in 2D, this is one of those graphs, and the problem of finding valid assemblies, by valid assemblies we mean symmetric assemblies with point group symmetry, how to find them, it is reduced to finding certain subgraphs out of these graphs. And we enumerate a series of rules, full coverage, uniform composition, and finite assemblies. So for instance, finite assemblies means that the subgraphs must be closed cycles. So there must be closed cycles in that subgraph for the assembly to be valid. With that, that's what we did in the version 3 of the EPPIC software. And we basically can now enumerate every assembly in the crystal, and we have a full, and this is an exhaustive enumeration of all possible assemblies, not only of the one, in this case, that we think is the actual, our prediction of what's the possible quaternary structure there, but we also have others. So for instance, there's the hexamer, but there's also 2 different dimers, a monomer and a trimer out in that crystal. So you get really a full analysis of that crystal. And you can also click on things here and get full the graphs of the interactions with color-coded types of interactions in there. With that, we did also a benchmark of and compare it to a very well-known method around PISA. And the result was we have an 84% accuracy in calling the right assembly. And the benchmark we used was by mining the whole PDB and finding all structures that have across their sequence cluster, so the homolog structures in the 70% sequence identity, that they have consensus, at least 3 members, and consensus of their biological assembly annotation. It's well known that there are fewer problems with the annotations in the PDB because of problems at the position time or ambiguities from the experiments. So that's why we have to have this kind of safe set. And this is composed by 1251 PDB entries. And this is the comparison where this is a confusion matrix of where this is the actual oligomeric size from the PDB, and this is the predicted one, and we have both EPPIC and PISA here in blue and red, and the purple shades are for the when both are present. And interesting here is the comparison across, so the value for accuracy for PISA happens to be 84% too, but if you really look at the details, this is monomers, dimers, trimers, and so on, you see here that there's across different types, there's different kinds of accuracies in both methods. So for instance, in monomers and dimers, we see that this is what both get right, and then there's blue for what EPPIC gets right, and red for what PISA gets right. And then, as you see here, EPPIC gets right more, and then here it's the other way around, so PISA gets right the higher oligomers. So with that, conclusions are that it's important to say again that proteins are symmetric, and there's rare cases where there's exceptions, and it's important to prove that those are real from experimental data. We presented this pairwise classification first, and then quaternary assembly prediction with EPPIC, which is 84% accurate. And importantly, we provide now confidence values, which can be very useful for the community to do large case analysis with this. With that, I'd like to say thank you, especially to everyone at RCSB Protein Data Bank. That's both in San Diego and in Rutgers University. Actually, most of the work was done while I was still at the Paul Scherrer Institute in ETH Zurich in Switzerland. And what I presented today is mostly Spencer and Alish who did. And of course, here I want to have a special big mention to Guido Capitani, who you heard the news. The group was really shocked for his sudden passing away this year. We really, so this project was actually started by him even before I joined the group in 2010, and we had the opportunity to work together all these years, and it was really a privilege to be able to interact with him, to spend hours talking about science. He had great knowledge and encyclopedic knowledge of science of all kinds, and I learned so much from him. So just some happy moments there in the group in Switzerland. With that, just to say that we have some open positions in San Diego, so if you're interested, please come to see me, poster 503. Thank you.
Thank you. Questions, please? I think that with the picture of Guido, we will all miss his brain that was struck by tumor, and we are happy that you're continuing the work on it. Any questions? And yeah. Hi, David.
Hi, I would like to know what is the equilibrium distance between in quaternary system? Do you have statistics, or are you able to depending on the group symmetry of the quaternary complex, there is some information about this?
So equilibrium does happen, and it happens more often definitely in larger assemblies where there's more possibilities to split them into sub-pieces, but we haven't really looked into that. We haven't really looked into, we know when interfaces are weaker, especially in interfaces that will produce sub-parts, sub-assemblies of the main assembly, but we haven't really looked in detail into it yet.
When you analyze the structures which are false interfaces, just lattice model versus the real interfaces, can you give any idea of descriptors, general descriptors of hydrophobicity or shape, which we can learn from just by looking without all of the more complicated evolutionary analysis?
So one of the things we look at, as well as the evolutionary part, we do some geometric analysis of that interface, and what we actually look at is a very simple measure, which is the number of fully buried residues in that interface. So the ones that, when the interface forms, their buried surface area changes 95% of to be buried. And that simple measure is really a very good indicator already, and we put that in our paper in 2012, where that alone will identify many of the biological interface. So that's sort of like the hotspots that, yeah.
Thank you.